Hello students, in the previous videos, we were discussing about the variation in case of Mendels law. Okay, and a few more variations are left and one of them is complementary gene. So we will start with complementary gene today. Now see the definition of complementary gene. The meaning of complementary gene is the gene which are located on the different loci of same chromosome or different chromosome, but they worked together to express one phenotype. Okay, see what I have told you. Complementary genes are the genes which are located on the different loci of same chromosome or different chromosome, but they all worked together to express one phenotype or same phenotype. Clear? See the definition. Genes which are located on the different loci of same chromosome or different chromosome. Suppose if I used to draw one chromosome like this. Okay, it's pair. Now, if the genes are located on the same chromosome but at different locus like C and P, then these are called as the complementary gene. Or what is the next term? On the different chromosome. Suppose if I used to draw one chromosome like this, okay, and its pair 1. Suppose if C is located here and if P is located here somewhere or here somewhere or here somewhere, then this is the different chromosome with the gene. This is the same chromosome but the different locus of the gene. This is called as the complementary gene. Okay, and this variation was discovered by two scientists. One is W. Betson and B-A-T-E and R.C. Punnett. Okay, these were the scientists who first time discovered the complementary gene and they worked on sweet pea. Okay, that is Lithyrus odoratus. So see what is the meaning of complementary gene? The gene which are located either on the same chromosome but at the different locus or on the different chromosome, then locus will be different. And this complementary gene was discovered by W. Betson and R.C. Punnett. And on which plant they have discovered this? And the plant was Lithyrus odoratus, that is sweet pea. Now see this complementary gene in detail. Okay, now why they have selected this sweet pea? What these scientists have found that these Lithyrus odoratus, that is the flower of the sweet pea, okay, of sweet pea was of two color. One was white and other was purple. Okay, and they saw that all the white flowers, when crossed with each other, that is, if white flowers were crossed with each other, then in F1 generation they found purple flower as a result. Clear? And in F2 generation they found purple and white both. But the ratio was not usual as it was in the Mendel's law. That is, the ratio was not 9:3:3:1. But here the ratio was 9:7. So this is the modification of the Mendel's law. That is, in case of Mendel's law, we got the result of 9:3:3:1. But in case of complementary gene, we got the result of 9:7. So we can say that it is the modification of the Mendel's law. Now, how everything has done, let's see this. Now, what I was telling you, that the color of the flower was purple and white. Now, just we can do a simple reaction, and the reaction is, suppose if we take the precursor. Now, the meaning of precursor is raw material. Okay. Now, with the help of enzyme 1, this raw material will produce a chemical called as chromogen. And this chemical is responsible for white color of the flower. Okay. Then again, there will be one more enzyme named as E2, and this enzyme will produce one chemical called as anthocyanin. Okay, and this anthocyanin is responsible for purple, or in your book it is written as red color, so no issue. Okay, so this is responsible for purple color. Now see children, these are the enzyme 1 and enzyme 2. This enzyme E1 is produced with the help of a gene called as C. And this E2 enzyme is made or formed from the gene P. Clear? Now see once again, what is the overall reaction? The reaction is, we have the precursor, and this precursor with the help of enzyme 1, and this enzyme is synthesized by the gene C. This enzyme will help in the formation of a chemical called as the chromogen, which is responsible for the white color. And then E2 enzyme will synthesize with the help of the gene P, and this E2 enzyme will help in the formation of a chemical called as a anthocyanin, that is also known as the pigments, chromogen and anthocyanin pigments, and this pigment is responsible for the purple color of the flower. Okay, now see what has happened. Now just see this reaction. Okay, for getting this purple color flower, okay, the enzymes or the gene should be in the dominant condition. If C will be in the dominant condition, then only the white enzyme or the white chemical will be formed. And if the P will be in the dominant condition, then the purple color will form. Okay, it means these two genes are complementary to each other. They are helping each other. Now see how they are helping. Suppose if enzyme C is not there, then E1 will not form, And if E1 will not form, then chromogen will not synthesize. And if chromogen will not synthesize, then how we will get the purple color? Not possible. So, this C is important. Now suppose, if we get the C, then enzyme E1 will synthesize and this enzyme will help in the formation of white color flower. Now suppose, this P is not there. If there is no P gene, then E2 will not synthesize. And if E2 will not synthesize, then once again the purple color will not form. That's why it is called as complementary to each other. Clear? I think so. It is clear to all. That is, these C and P genes are complementary to each other. They are fulfilling each other's requirement. Clear? Now see, what can be the situations? Now everything should be in the dominant condition. Now what will be the conditions? We all know that maximum we can have the 4 conditions. Like, if I write here, then is it possible to see? Suppose if C and C, this gene responsible for the E1 and P responsible for the anthocyanin, that is responsible for the E2. If they are in dominant condition, then what will be the color of the flower at the end? The flower will be purple. Clear? Now, if this C is this C, P and P, if they are in the recessive, okay, then they will not express themselves. And if they will not express themselves, then definitely the color of the flower will be white only. Clear? So, not purple. Now see the next condition. The condition is, if there is one C capital, one G, that is one allele, and another is in the recessive, again P is capital and P is recessive, then at least we should have one allele as a dominant. And if we have one allele as a dominant, then the color of the flower will be purple. Because here at least we get both the genes in the dominant condition. Now see the next one. Suppose if this is capital C, this one is in the dominant condition and this one is in the recessive condition. Now what will be the result? There will be the formation of chromogen. There is the formation of white flower. But because P is in the recessive condition, so there will be no purple. Clear? Now see, once again, one more situation. Small c, capital P, capital P. Now if this is in the recessive condition, then it is not going to form the chromogen. And if chromogen will not be there, it means if intermediate raw material, if intermediate precursor is not there, then P is of no use. And once again, the flower will not be purple. Okay, now see this board carefully and see what I am telling you. I am simply telling you that just to get the color, okay, both the genes, complementary genes, should be there in its dominant condition. Clear? Out of the two, if anyone will be in the recessive, then we will not get this color. So this was discovered, everything was found out by our two scientists, W. Betson and R.C. Punnett. Okay, now let's have the cross of all these things. If I'll draw the cross, then I will show you how the ratio is 9:7. Okay.